it's usually in gaseous molecules or maybe something like dye, or it could be heavy mass of gases. So it has a net movement. Okay, the key thing is it has a net movement of particles from a high concentration gradient to a low concentration gradient. And this is usually result of their random movement. Okay, this is the definition that by now you should know what it means. Okay, by now you should know what it means. Okay, so for this session, I have, if I have not told you, be about 1 hour to 1 hour 30 minutes. If I can get it done, then that'll be awesome. We will still have a Q&A session and a tip session before we sum up everything. Okay, all right, let's get back to it. So I will show you one diagram of how this works. So for example, if I have a beaker, okay, and I assume that this is distilled water. Okay, maybe I have a like a drop of maybe like some blue color dye um into this beaker, and it's going to be dissolved into, not really dissolved, but then it's going to start to mix in together. So what happens is that how diffusion works is that it starts to spread out, because they do not have like a fixed shape, right? They won't be staying static like that. It will move and spread out. So for example, if this is blue color, right, the whole entire solution will become blue in color. So this is called diffusion, something like that. Okay, another way is that maybe if I have how would I say, that one side I have a lot of these gas particles, and I have one side of it which has only maybe three of it. So diffusion happens from a higher concentration gradient to a lower concentration gradient. So this like this extra gas particles you can see, it will eventually diffuse down so that it can move from a higher concentration gradient to a lower concentration gradient. So what is the concentration gradient is that there is the both sides have different concentration. That's what I mean. Is that one side may have more, one side may have lesser in amount. So when there's higher amount, it will always flow down from higher to lower. It's always like this. Okay, now we'll enter osmosis. For osmosis, it will always involve water molecules. Now we, okay, to not confuse you guys, we will take water and some salts. Okay, when we have a high amount of salt, we call this as high in solute concentration. So when you have highest amount of salt, you're going to have high solute concentration. All right, I think you should get this part. So when there's a one side, right, there is a lot of the water molecule, and then one side has lesser water molecule. And maybe this side, right, it has more of this maybe sugar, or maybe there's a lot of salt that it has a differing gradient from the one next to it. So what is going to happen is, right, these many water molecules will in fact move down from a higher water potential to a lower water potential. So this is called osmosis. So how would you structure your um, definition? So it will be the net movement, okay, of water molecules. Okay, apologies for my handwriting. I try my best because I'm also trying to not drag this too long. So net movement of a water molecule from a higher water potential. Okay, why I would say higher water potential? Because there is more. That is potential, meaning there's a lot of that thing. So what is happening? There's a lot is that it starts to move down to a lower water potential. So from higher to lower water potential. Water potential. And one more thing, across a partially permeable membrane. So what do you mean by this term of partially permeable membrane? Is that partially permeable membrane is, it's a thin layer of membrane where a lot of things can easily penetrate into. So it's usually, for quite a small molecule, water is quite small actually. So therefore it can, move down its water potential easily. Okay, so large molecules like protein, red blood cell, white blood cell, they cannot pass through this permeable membrane. I think it really differs now. Sometimes it may, sometimes it cannot. It depends on the. So down question, what's the difference between? They are the same thing. Semi means half. Okay, let me put that word. Semi is equals to half. So partial is also equals to half. It's a synonym. Okay. Mm. Okay, so same thing. All right, now we'll move on to active transport. So active transport works slightly different. Why? Why? Because most of the time, right, it's always from higher to lower. But this time, active transport is going against it. So it's from a movement. Okay, I write the definition down. Movement of particles from a lower concentration gradient to the higher concentration gradient, or some, if you know the concept, is against the concentration gradient. So I would say against the concentration gradient. Okay. And one more thing is that it using energy from respiration. Okay, so this one is very important. Okay, now you'll be asking me why you know this active transport works so differently than the osmosis and the pass and the diffusion. Why? Because now they are using energy. Energy is the one that helps it to go against its concentration gradient. By law, it shouldn't do that. By law, it's always from a higher to lower region. But this time, because they have energy, this energy will help to pump across the concentration grade against the concentration gradient. So that is why active transport is so useful in a lot of our mechanism, and including the plants, when root starts to absorb it, then it begins to undergo this active transport, and they can absorb this type of nutrients, okay, during in need or maybe scarce in resource like that. So this is how active transport. So active transport will of course have a protein. Okay, they always have a protein on the membrane, and this protein can help to shuttle these molecules they want to go to other side from a from a lower concentration gradient to a higher concentration gradient. All right. For plants is silent, but flowing it moves up and down. So what's the mechanism in the phloem? Okay, I will come to that later on. So we go step by step. Okay. All right, now we will enter chapter 4. Okay, so for chapter 4, this will be the food test. I just want to recap the food test with everybody. So make sure that you must know this. 100% is going to come out in paper 6. There is there's no way to avoid this. Okay, so let's refresh for food, and also paper 2. They sometimes can come out. All right, so the first thing is we are going to test for this reducing sugar. So reducing sugar can be glucose, okay, glucose, fructose, okay, all those monosaccharides that can be tested for this. So the reagent that we use is a Benedict solution. Benedict solution and Benedict reagent is the same thing. Okay, for a process, okay, a process we use is by adding the solution, okay, adding a solution, and then we heat up to about 70 to 80 degrees. 60 is also acceptable. So just around that range. Okay. So the positive test for reducing sugar will be break rate, and it is a negative test, it will remain as blue in the precipitate. All right. For the next one, we're going to test for starch. Okay, so starch is a very long chain sugar. So we use iodine solution to test for this. So iodine solution, we just add it. There's no specific condition it has to require. So the positive test, it will be blue black in color. So any color that is similar to this, it should be acceptable. Just make sure it's not some other random colors like yellow or red. And the negative will be brown. Okay, because iodine itself is brown in color. All right, the next one is to test for protein. For protein, we will use a burette solution. So for burette solution, we also add it. There's no specific condition we need to use. And of course, the positive test will turn purple or maybe violet. Okay. And the negative test is that it's still gonna remain color. All right. And the next one is to test for fats. So fat is rarely asked, but then just know it as your reference. And fats, we use the ethanol or any ethanol is the best typically. So the process is just to add the ethanol in, then we shake it, and some may say shake it with equal volume of water. Okay, you can just add it and shake it, that's enough already. So the positive result will be a cloudy emulsion. Okay, and the negative result, there's not really a negative result for this. It's either it occurs or it doesn't occur. Then lastly is for vitamin C. Okay, vitamin C we use DCPIP. Of course we just add the DCPIP in, and then the positive test will be colorless, and negative test will be in blue. All right. So simple. I would say that this is a simple one. Okay. So now we enter DNA. Okay, DNA. Please remember this, that A binds with T, C binds with G. So there are some calculation questions which relates to okay, example that A has this amount of bases and how many bases will this G might have, something like that. So make sure that you know that the amount of A is equal to the amount of T. The number of A bases is equal to T bases. So it's always equal. All right. So that's it. Very simple. Okay, now we enter chapter 5, which is enzymes. So far, are there any questions? Would you like to ask? Any questions? Yes, I will open the floor for people to ask questions now before we enter chapter 5. Oh, check Google Docs. All right. Oh my god. Wait, where is it? Wait, biological. So, guys, should I just paste the thing I made? I think sure. Can. Wait, let me check any of the questions here. Synapses, gas exchange. Okay, so far nothing about chapter 3 and chapter 4 yet. All right. Is it fine if they can explain active transport? If I don't mention heating up, this kind of question will come across in paper 6 if they ask you for how are we going to conduct the testing of this reducing sugar. You must mention heat. If you don't mention heat, it will be a deduction in marks. Okay. All right. I will explain active transport. Quick one again. Uh, there'll be a recording. Just hold on. They will the mods will send in. Okay, for active transport, remember it's going against the diffusion and the osmosis. Why? Because it's going from a lower concentration to a higher concentration. So when it moves from a lower concentration to a higher concentration, it will need to use energy from respiration. So this must be a living thing. The living thing must be able to undergo this cellular respiration, and when they produce this energy, they only can undergo this active transport. So active transport is very good in dire situations of a particular organism when basically they want to need some nitrate ions, for example, and the surrounding is very little. But no matter what, because they have the energy to do so, they can use active transport to move from a lower to higher in concentration gradient to still get that nitrate ion regardless of whatever happens in the surrounding. So this is a very useful mechanism. So just remember, active transport is going against the concentration gradient. Okay, awesome. All right, now let's move to chapter 5, enzymes. Okay, for enzymes, I the first part should be very simple. The lock and key model. So we always have an enzyme, and we have a substrate. Okay, so this enzyme, right? It has an active site. So this active site, what is the purpose? Is for a substrate to bind with it. So remember, right, this lock and key model, it needs to be exactly complementary. So take note, it has to be exactly complementary. So for example, this lock and key model is derived from, for example, I have a padlock, okay, and I have a key, for example. So this key, right, in order to unlock the door, the shape of the key has to be the same thing, the same exactly complementary as the lock itself. So only this can undergo the unlocking process. So this is the same concept as this. The substrate will act as the key, and when it binds with the active site complementary, it will form the enzyme substrate complex. Okay, so this is the basic concept for this lock and key model. All right, now we will enter the temperature curve. So this type of question, it has been asked many times in paper 4, paper 2, especially in paper 4. They will always give you a situation, and this enzyme works in this way, and they will ask you to describe and explain the curve. So they will always ask you to describe and explain this temperature curve. So it always gives you maybe 4 to 6 marks. If it's a describe, it may be 3 marks. If it's a describe and explain, it can go up to 6 marks. So first thing, when you come to this kind of question, you have to strategize your response. So how are you going to strategize this response? Is by breaking down. Describe is by describing the shape of the curve, and explain is by explaining why when this curve increases, then um, this thing will happen, this curves will happen. Oh yeah, wait, hold up. My bio teacher mentioned something about a competitive and non-competitive inhibitor. It's not inhibitors, it's inhibitors. What are those? Not important for the syllabus, but if you want me to explain later on. It's A levels. Okay. Um, so how am I going to explain this? Is like initially I will start with this point. I will take first point, second point, and third point to explain. This will help me to aid my explanation. So initially, at lower temperature, there will be less kinetic energy. Therefore, there will be very little of this enzyme reaction occurring. When temperature, okay, now I'm coming to this part. When temperature increases, what happens is that the molecules will begin to move faster. So why it moves faster is because of the kinetic energy. When the kinetic energy also increases, there will be more and collisions, Okay, and we can also say frequently collide with other particles, then to form more of the enzyme substrate complex. So more enzyme substrate complex. So this is some key points that you can use. Then when it reaches the optimum temperature, you must always state what is the optimum temperature and how does this affect. When it's at its optimum temperature, right, they work at the best. So the percentage activity of this enzyme will work at the very, very optimal level and will be the fastest. So there will be the most, okay, most of the products will form in that reaction, in that optimum temperature. And what happens is that when, okay, yes, there will be this later on, okay, menstruation hormones later on. I will explain this properly, okay? No worries, we start from the beginning to the back. Don't worry, all right? Okay, and the third part is that when the temperature further increases, maybe it could be 46 in degrees Celsius. So what happens is that the kinetic energy will begin to decrease. And what happens when this kinetic energy decreases? It slowly changes the shape of the active site. The shape of the this important because that substrate is unable to bind exactly complementary anymore. Because of the active site change, the key cannot, bind with it anymore. How does kinetic energy decrease? Sorry, not kinetic energy decrease. I'm so sorry. Is that when kinetic energy further increases, this causes more vibration and the distortion of the structure. Apologies, apologies. So sorry, Okay, so what happens is that I'm sorry for that misunderstanding. Okay, basically what happens is that when too high of the temperature, right, this thing will start to, you know, shake, vibrate more too much, and then this thing will begin to change shape because the protein structure, enzyme is a protein. Remember, enzyme is a protein. So when enzyme is a protein, right, when temperature affects it, right, it will cause the distortion of the structure. That's where it brings to this point. Change the shape of the active site. Okay, once it changes the active site, that's where we meet this. It finally denatures. Please do not write die. Okay, the enzyme die. The please is supposed to be denature. Okay, all right. Yes, I am. Okay, now pH curve. pH curve is the kind of yes, yes, it distorts the entire structure because protein is very sensitive. Protein is very, very sensitive. It's almost like us as a human body, right? When too high the temperature, right? That's why people get the heat stroke and everything, right? So that's why it affects our protein. We got a lot of protein. Our muscles are almost built with protein. Hair is made from keratin. It's also protein. So all this can affect. So that's the reason why. Okay, for pH curve, okay, same thing. There will be always be one side, okay, that has optimal pH and another enzyme that has another optimal pH. So for this is slightly different. Why? Because before its optimal pH, right, the percentage activity is very low, very low. So at lower pH or before, I think I don't say lower pH, before optimal pH. I think that's a better keyword. So when it's before optimal pH, okay, there will be lesser enzyme activity. And when it reaches this point, is where enzyme will work best at its pH, peak performance. Okay, I always call it as peak performance. Then what happens is that when it reaches peak performance, what happens? When the pH again changes, it drastically drop. Okay, this curve, right, drastically drop the moment it starts to go further away from the optimal pH. So drastically drop in the enzyme activity. If you wonder why, same thing. Protein, right? One thing, the factor is temperature, correct? When there's too high the temperature, the kinetic energy will distort the structure. That is one thing. The second thing is pH level. Our proteins, our enzymes, they are also very sensitive to this pH. So what happens is, right, when the pH changes, the bonding in the protein will start to break down. When it starts to break down, that whole shape of the active site begins to change. And the same concept is that it cannot be exactly complementary. Okay, so one thing is going to happen is that this whole thing will be denature. Okay, so if you're wondering why am I explaining so much, is because I want you to know why does this happen. So why does this happen? First of all, no matter what, if you're not pursuing biology in your next stage, um or maybe in your future, it's important that you understand why, because A level is surely gonna ask something like this. Okay, so that's very important. All right, make sure you understand this. Okay, so any questions? Any questions from enzymes? Let me see. Are there any questions? So far, don't have. Okay. Okay, that's nothing. All right. What's the difference between the enzyme denaturing from temperature and from pH? Uh, there's no there's no difference. The same thing. It's just that it has two factors. The two factors are temperature and pH. That's why we have a temperature curve and we also have a pH curve. So when this temperature and the pH exceeds is this what you call as this optimal condition, it starts to denature. So it's still the same thing. Both will change the active site. Mm, okay. All right. Awesome. Okay. Now I will enter chapter 6. So chapter 6 is a hydrogen carbonate indicator. So you will see this kind of question in paper 6. They also maybe in paper 2, saying that this particular plant is in this condition and that particular plant is in another condition. Okay, wait. If you want to ask any question, here is the document. Okay, for you to enter your questions in here. Make sure you check in the chat. I will go through methodically later on. All right. Okay. So how do I remember this? So you don't panic when you see this kind of question. I always love to use PM Roy. So um yellow is being the highest in that carbon dioxide concentration. He being the lowest carbon dioxide concentration. And red is usually the center, or maybe orange. Okay, so remember this, okay, because definitely gonna ask you some questions on paper 6 maybe, or maybe paper 2, and this kind of condition of like when the plant is trapped in a particular test tube where there's no sunlight, okay, there's no exposed to that particular condition, how would that hydrogen carbonate indicator may change, something like that. All right. Okay, now we will enter limiting factors. I'll just go through a little bit only, not so much. Okay, so what happens in light intensity? So plants, right? They are, remember in the photosynthesis equation. Okay, let me write down 6 CO2 plus 6 H2O C6H12O6 plus O2. Okay, in the between is light and chlorophyll. Okay, so what happens is that light is very important. If there's very low light intensity, the rate of photosynthesis will be lower. Because for example, if this is 10 and maybe this is a 50, this is 100. So when there's maybe very low of that light intensity, very less in that sunlight, see the rate of photosynthesis is just 10. Okay, but then as the light intensity increases, it can eventually reach about 50. So that's how light intensity plays. But then you cannot keep on adding more light to that plant, because remember, photosynthesis is also the involvement with enzymes. So remember that enzyme is also very sensitive to temperature. So when enzyme, of course, when this light intensity provides too much of that light, it starts to burn the plant. The plant will begin to burn. That's why to protect that, it remains constant. It remains stationary. Okay. Okay, any questions, do drop in my DMs. I will try to answer, okay? Maybe not so soon. I try to do it before I go to sleep. Okay, next one, CO2 concentration. Very important. CO2 is a reactant of photosynthesis, right? So it's important that when we have a lot of CO2, the rate of photosynthesis will also increase. That's why at a lower CO2 concentration, there will be a lesser rate of photosynthesis. But when this increases, it can become higher. But then eventually it will also be remain stationary. Why is that? Because too much of the CO2 will also cause the acidity. So when there's high CO2, the pH will drop. So this concept is the same thing in our lungs. Remember, when there's too much CO2 in our blood, the pH drops. So we do not want that our body to become acidic, because the moment it becomes acidic, we can actually have problems. So this is the same thing for plants, okay? And temperature. Why doesn't the graph? It did drop down. It did drop down eventually. It's just that it never show. It did drop down. Confirm will drop down one. All the graphs, by the way, this is the extension of the graph. But we just show this is just to make sure you understand that at some point it has to stop. You cannot add too much. And both proteins are sensitive to both pH and temperature. Mm. It's something like we as humans, when temperature is too high, we get fever, correct? When we got 40 degrees, when the temperature was like 3 degrees above our body temp, why is it eventually drops? Why it drops? Because remember light intensity, too much of that light, it will burn the protein. They will burn the enzymes in the plant. Therefore, they are unable to undergo photosynthesis anymore. So the rate of photosynthesis will drop. At some point, it links with temperature, and CO2 will link to pH. Something like that. So is this sort of like a negative feedback? Yes, correct. It's like a homeostasis mechanism. So it's that's why it's the same as us. We living things, we always have a homeostatic mechanism. Whenever something just goes beyond our optimum condition, right? The body or maybe the organism itself will try to pull back itself, go back to its normal state. Mm, that's why we cannot have too high fever. The moment we get high fever, right, a lot of our body functions will all go haywire. So that's same thing. Okay. So for temperature, it's the same. Can you see but high fever can, yes, but then it's just you cannot have too high of the fever. Not have too high, because temperature is the one that will affect. Okay, how's the CO2 linked to pH? Okay, let's take this example. Our lungs, right? When we store too much of the carbon dioxide, and then what happens is that this carbon dioxide will bind to the blood, the hemoglobin in our blood. So what happens is that this will affect the transportation of these gases to our tissues that are trying to undergo respiration. Okay, so that's the concept. Do enzymes corrode to too low pH? Don't say corrode, but it will denature, or it may distort the structure. The structure will change. Can I be uh, at some point if it's too high. Mm. Not our enzymes. Yeah, our enzymes is unable to function. It's unable to function. But if it's pepsin, you'll be fine. Yes, yes, yes. See, pepsin cannot undergo alkali condition, right? It only can goes well in the acidic environment like that. Mm. But then okay, don't worry, let's don't go too far. So temperature is the same thing. It's almost the same as this temperature curve when there's mm, why does it plateau? It plateaus because like CO2, they cannot I mean for the light, right? They cannot have too much because it will burn the plant basically. Just think of it will burn the plant. Okay? And then CO2, too much of the CO2 you make it acidic. What if we don't have enzymes? A lot of things cannot happen. Don't have enzyme, everything cannot happen. Mm. Okay? Plateau means goes to stationary. It becomes stationary. Basically, it just curves, curves off like that. That's it. All right, okay, let's move on. Otherwise, we do not have enough time. When curves just stop changing, yes, that's true. Can we eat enzyme? No. Enzymes, do we eat enzymes? Mm, no lah, no. Do different enzymes have different? Yes, certainly, correct. But um, but for our human body itself is set this way, it sets this way. Mm, okay. All right. Now let's undergo chapter 7, which is the human nutrition. Okay. So previously we talked about plants, and then now we will talk about this human nutrition. All right. So what are the key things you have to know? There are so many things that happens in the human nutrition. It all begins with ingestion. So I always take ingestion as taking substance into our beginning of the body, which is the mouth. Okay, into our mouth. So if you don't remember, in means going in. So going in our body is by eating something. Okay, that's a very simple example. Now this will be mechanical or maybe physical digestion. So what does this mean? Mechanical means we do not use any sort of enzymes. Okay, we do not use those bile or those to to digest any of it. This is more of how the body is just doing its job. The structure of the body that helps to to digest our food like that. We don't break bonds in. Uh, no, we don't. We only increase the surface area of it only. Mm, okay. Like teeth, basically teeth. Our tongue also helps it. Okay. Mm. All right. Now, this is mechanism. I just say that is the breakdown of food to smaller pieces. Okay. Now, chemical digestion. This is the part where we use that enzymes. We use the bile, so we break down large maybe insoluble molecules to small and soluble. So these enzymes, right, they will start to break the bonds, basically. Okay, they will start to break the bond. Okay, churning, yes, peristalsis, they are all considered as the mechanical digestion. All right, okay. Then next we will undergo the absorption. So absorption happens with after everything has been digested into a smaller pieces, and now it's soluble inside the liquids in our body. It starts to absorb in our small intestine, start to absorb into our body cells in the intestine. Small intestine, to be more specific, because the villi are present there. Okay, then we have assimilation. Assimilation is like, okay, after everything has come into our body, okay, how everything is going to come in now, okay, the body will start to use these what do you call, they will start to use all of the nutrients that it has been absorbed inside to make something in for our body. For example, muscles. The more protein we eat, the amino acid that gets absorbed into our body, assimilation will occur. It means that the body will use that amino acid to make the protein or make the muscle to be more specific. Okay, so the assimilation is, yeah, so the use of the nutrients make new cells. Okay, use of nutrients by the cells. Okay, and lastly, egestion. So egestion is to remove undigested food from our body as feces. Okay, so how do you remember egestion? Egestion means exit. So it exits our body. Okay, so now let me ask you one question. Okay, which particular nutrient, okay, um, that cannot be digested in our body? Which? Correct. Okay, fiber. All right, so fiber is the only one that cannot be digested and it just straight away enters into our feces. Okay, all right, I think I have one question. Where does assimilation take place? It takes place everywhere in our body. Because our body will undergo this kind of like process of protein synthesis. You know protein synthesis? Ah, okay, protein synthesis. So all of our body parts are able to undergo this. All right, I think that's a good question. What's the difference between egestion and excretion? So please do not confuse between these two. Egestion is poop. Excretion is pee. Yes, that is the two things. Because excretion, right, is you're getting rid of the waste product. And that waste product is usually in that liquid form. But for feces, right, it's waste material. Maybe those are undigested food is one example, and they cannot dissolve any further basically. So therefore, it releases out in our body. Yeah, maybe like excreting carbon dioxide, or maybe urine or sweat. What do you have the exploding diarrhea? Okay, diarrhea happens, fun fact, diarrhea happens when the body cannot absorb the water. So something when water is not reabsorbed, right, means all the water is basically washed into our large intestine, then enter the rectum. Therefore, our diarrhea is become more watery type. You get what I mean? So this is a very good application. If you ever wonder why this happens, is that you know that water just never gets reabsorbed. Yes, we have cholera later on, so stay tuned. Okay, now, oh yeah, sorry, I think there is some stuff that did I miss anything? Okay, so the mouth, okay, the main thing is mechanical digestion. So this is the mechanical digestion happening. Okay, salivary glands to secrete the saliva. They secrete the saliva, or maybe the amylase that helps to digest the starch. Esophagus is where the peristalsis movement occurs. So this constant contract and the relax of the muscles that helps to move the food forward and not going backwards, Okay. And we come to the stomach. The stomach has the pepsin, okay, which is the acidic protein enzyme that helps to break down the protein into an amino acid. And also the stomach has a very interesting wall that helps to prevent the hydrochloric acid from dissolving the stomach layer. So that stomach has a particular protective layer that helps to um, to digest it. Yes, mouth can undergo both of the mechanical and the chemical digestion, I would say, because the chemical digestion occurs in the salivary glands. That's true, correct? Just that the main thing, the mouth, is that it has the teeth, okay, it has the tongue that helps to chew the food and increase the surface area. Okay, so what do we need to know? Which one? What's the protective layer called? No need, no need, no need, no need. Don't need to know. I don't want to put too much thing in your brain. Okay? Do not need to know. Okay, and we have the liver. The liver helps with the deamination process and also the detoxification of blood. That's where all the alcohol is being broken down and the production of the urea, which is sent to the kidney later on. Then we have the gallbladder. The gallbladder will store the bile. They do not produce bile. Very important. Please do not confuse it. Pancreas will produce the trypsin, the amylase that are very good in that alkaline condition, and also lipase sometimes. We have the small intestine. That's where the microvilli, the all the digestion also happens there, and the absorption also occurs. Large intestine is where um, they also helps to reabsorb some of the vitamins and the minerals, um, and then to maintain the water levels in our entire body. And the rectum, which is here, is where the poo poo is stored. Okay, so the rectum is where all our feces are stored. So the gallbladder stores and see. No. The liver will produce the bile. The liver will produce the bile, and the bile is stored in the gallbladder. And when there's fat molecules coming, then it secretes it. That's a different thing. Trypsin and bile is both alkaline. Trypsin and bile is both alkaline. Yes, correct. I don't yeah, just to recap only. Sorry, my bad. Okay. Uh I will explain epithelium later on. Hold up, okay. Trypsin is an enzyme. Yes, yes, I think he means that trypsin works in the alkaline condition. Enzyme is also a protein. Don't worry. Okay. Okay, now we got this teeth. Okay, for teeth, right, you of course must know what's an incisor. The incisor is the front of our mouth, okay? And the canine is just next beside the incisor. So we're going from front to back, by the way. So the front will be the incisors, which helps for cutting. Okay, so I just write there the incisors helps for cutting and some form of biting. And we have the canines, which is for piercing or maybe for paring. And the premolar is about this size, okay, which is for chewing. And molars are also the same thing. They also chew because it's from the back of the teeth, right? So it starts to help with the chewing process. Okay, all right. I'm worried that I got so many questions. All right, no worries. These are all later on. All right, I need to go faster, guys. Okay, so the the animal, okay, this layer, right? We always see the white color layer in our teeth. These are the strongest tissue in our body. So just for this part, just know the label. Know the labelings. You do not need to know the function, just the labeling. Okay, know the labeling. So the dentine is here. The pulp is where the veins are. Not the veins, the capillaries. Then we have the cement, okay, which is located here. The root, where it attaches with our gums, and the crown. Do not need to know. Just know the animal. Dentine. This is based on the syllabus, by the way. Pulp. Um. Semen, yes. That's it. Okay. Then just know that these are all the blood vessels. Okay. All right. All right. Okay. Oh yes. So this is chemical digestion. So we go through some of the enzymes that happens in here. So these are the enzymes, and this is the function. So for enzymes, we have amylase. So it's to break down the starch into the maltose. Yes, starch into maltose. Then we have the maltase, which is the breakdown from maltose to glucose. And we have the protease, which we can either have pepsin, which is located in the stomach, and we have the trypsin. So they break down from protein to amino acids. And what's next? We have lipase, which is from fats to fatty acids plus glycerol. Not glycogen, by the way, glycerol. Why do body need trypsin and pepsin? Two different conditions. Because the body unfortunately cannot digest so much protein in once. So when example like you go for a party, you eat the buffet, a lot of things in your stomach, right? Your stomach has a capacity. The stomach will try to um, digest as much of the protein as they can, and at some point the stomach has to release the food, not the digested food. Then the trypsin will do the further breakdown. When they do the further breakdown, then yeah, that's where it starts to become the amino acid fully. Then it will be reabsorbed back into our body. Okay, all right. Coming back, then we have last one. I think that's it. Yeah, that's it. Okay, now what is the role of the hydrochloric acid? So there are some few past year paper questions that ask what is the role of the hydrochloric acid. So first thing is to denature enzymes in the microorganism. And secondly is to kill the pathogen. And the third one is to give the optimum pH for pepsin activity. All right. So these are the three. Oh my god, 22 more left. Oh shit. Okay, sorry. Okay. Okay, let me know, right? If you oh, there's a lot of things there. Antibiotics and bacterial resistance. Okay. All right, all right, all right. Okay. Oh, there's a lot of questions too. Small intestine. Yes, yes, yes. You do not need to know erapsin, by the way. Okay, come. Let's tell you about some few good terms to know. Okay, when you eat the food, right? Okay, the food is in the mouth. Sorry. So this is the food. Okay. After it has been digested already, right? It becomes here as a bolus. So bolus is the type of that big, this kind of a small ball of this kind of digested things. So this is after it has been um increase the surface area, the mechanical digestion has occurred. Okay, then what happens is that it enters the stomach. So after the stomach has already do the dissolve and everything, erapsin is not in the syllabus, by the way. Please do not worry about erapsin. You need to know for your syllabus. Okay. Okay, then after the stomach has secreted, okay, that stuff, right, it becomes chyme. So chyme is a liquid. So that liquid is gonna go everywhere, okay, to our large intestine and to our not really our large intestine, but mainly our this small intestine. That's where all the chyme is. No, this is you do not need to know what is a chyme. I think this is just for understanding purposes only. Okay, just to make sense. Okay, that we have a different terminology for different kinds of process of the food. After it's been broken down, after it's been digested, they always have a different name because the form has been, modified already. Don't worry so much. Don't worry so much. Okay, then we have bile. Okay, so bile will undergo this emulsification. So remember, liver produces it, the gallbladder stores it. Okay, and this bile will undergo this emulsification. Okay, so this bile, right, it has this an alkaline condition. So this helps to break down the large droplets, okay, the large fat droplets of the fat into smaller droplets. This is all for the ease of that absorption, okay, later on, okay, so that the lipase can also further break down. So this bile is just to help to, what's the pH of bile? Alkaline condition, uh, I would say maybe pH 9, pH 8 to pH 9, slightly higher. You know why? Okay, let me tell you a story. Okay, there was once that I had a massive nausea after eating something that's not right, right? So I kept on vomiting. So that vomit, right, was at first was all the food. Then I ate some medication. I still vomited the food. I still vomited the medication out. Then at some point, right, uh, I started vomiting some yellow liquid. Then I was wondering what is that yellow liquid, and that yellow liquid was in fact the bile. So when you start to vomit out that bile, right, it tastes very bitter. That bitter rich taste shows that it's alkaline. No, no, no. Because this was like a few months after exam already, and then I tried to mm, learn something different. I was like wondering like what is this substance that I was vomiting because of course because it's interesting. I'm a bio fan, so that's why it tastes that some of like the bitter type of not bleach, totally not bleach. It's just that very bitter kind of taste like that, which was very disgusting. I didn't vomit on purpose. I ate something wrong. Maybe the food wasn't cooked. I think because I was so I do not know. Because you can smell bleach. Bleach doesn't have that sort of that smell also. So something different. So at the time when I was vomiting bile, I was like, oh shit, something didn't went right because my whole throat, not my whole throat, my whole esophagus was burning, because esophagus is not meant to have that sort of like that acidic and that alkaline condition, to occur, So that was very painful. So yeah, that's the story. Okay, let's go back. So now we come to villi. Okay, so, am I still speaking? Can you still hear me? Okay, all right. So now we come to the villi. So the villi is very important for absorption. So absorption is key because whatever we have digested, it has to go into our body. Our body needs this valuable resources. So this we have a lot of villi. So in for example like this particle lining, right? There's a lot of the villi. So you'd be wondering why. Oh shit! Why this thing just delete itself? Something. Okay, never mind. My drawing very bad. So it's to increase the surface area. So many of the villi will help to increase the surface area. Okay, that's very key. So that the absorption can happen. All right. Now we look at this epithelium. Okay, epithelium. I think somebody asked me what is an epithelium. Epithelium is what do you call as um this it has this kind of cell. Let me show you how the cell looks like. And if you look closer, this this what we call as the epithelial, right, has a lot of the microvilli. Okay, something new that you guys may not have known is that this structure is a villi. But a microvilli is on one of these cells. So many of the epithelial, right? And each of the epithelial is lined with many microvilli. That will increase the surface area again. So, okay, now back to here. The lacteal will help for absorbing the fats, okay, the fatty acids. The capillary is the blood vessel. do not need to know this. The then the artery and the vein is just to what do you call it as like um to oxygenate it, something like that. Just the normal thing that it happens everywhere in our body. All right. Okay. Um, boring part, but bear with me. Okay. Um, so for this one, make sure you know the structure. This is a leaf, this is a stem, this is a root. So you must be able to know which is the phloem, which is the xylem. So how do I remember this? Is that the leaf, right? It has a triangular shape always, and I always know this is a xylem. And this kind of shape looks like not to say a very nice semicircle, but this is a phloem. Okay, so I hate plant, don't worry. Okay, stem is easy. The top part, this is the phloem. This part is the xylem. The root, the X shape, is the xylem. This part is the phloem. Okay, so just understand this. Okay, do you want me to explain transpiration? Do you want me to explain transpiration? If no, I can skip. Yes, all right. I thought you said plants are boring. Okay, never mind. Okay, let's um let's do transpiration. Do we got to memorize the monocot, dicot? Okay, this plant is monocot. This type of plant is monocot. Okay, this type of plant is a monocot plant, typically. Okay, so how does transpiration occur? Okay, we apply back our understanding with osmosis. Yeah. Okay, so when water is being absorbed into our into the roots, right? It goes from a higher water potential to a lower water potential. So it enters the root hair cell. So after it has entered the root hair cell, what happens is that it's gonna start to meet with the cohesion. Okay, the cohesion means between the molecules itself. Okay, it has that sort of like um attraction between it. So the water will move upwards. Okay, the water will move upwards definitely, so that it can go to the rest of the leaf and all those. So it's known as the transpiration pool. You know why? Because when we're going up, we are going against gravity, right? So we need to have a certain kind of force that helps to pull the water up so that it can go to the different organs in the plant. So this is the transpiration pool. Okay. Then what happens is that it will draw a column of water molecule. So meaning that water molecules are almost lined on the wall and it goes up. So that lining up is the cohesion. Okay, so it has a water molecule has a itself right has a very good cohesion with the walls of the plant because it's a very strong bonding. Okay, remember, so it's a very strong force of attraction, something like that. So as it goes up, then it will go to the leaf, all those. Okay, then after it's been used up, then it leaves as a water vapor. So this H2O will become the gas water vapor. So how does this what's the definition is that transpiration is the loss of water vapor uh, which of course they undergo the evaporation because it's a gaseous state now, uh, which is out from the air spaces, evaporates out from the into the air spaces by the diffusion. Okay, so whatever the H2O gas leaves, right, it's all by the diffusion. All right, so apply back the concept that we learned just now. So yes, it was about 2 to 2 marks, I would say, not three. Three is a bit too much. Entire process 5 to 6, decent actually. But then, typically, I don't see a lot of this. But then, I hope Do we talk about the adhesion and cohesion? Yes. You can mention this too. The adhesion and cohesion. Yeah, cohesion is the main one. Cohesion is the main one. Hmm. Because cohesion is the one that the water molecule will stick onto the stem of the plant, then it goes up by the transpiration pool. Will you get marks for answer? Yes, definitely you get marks for cohesion and adhesion. But make sure you explain that it lines on the wall. Um, of course, because it has that strong forces of attraction with it. Cohesion is the attraction between water molecules. Yes, correct, correct. Something like that. Something like that. Oh wait, my bad. Cohesion is between water molecule and water molecule. Adhesion is between yes, water and the wall. Okay, make sense? Mm, yes. Correct. All right, awesome. Good job. So that's it for this transpiration. Do I need to explain translocation? No, just right key points. Okay, translocation is involves the phloem. Okay, so always from the phloem. Then you transport the amino acid and the sucrose. Okay, and of course from the source to the sink. All right, that's it. Very simple. Okay. Adhesion. What I mean is the water and the wall. Okay. For example, right. Okay, this is the xylem wall. Okay. Then this water molecule, right? They will line on the wall. This thing is called adhesion. It's the water molecule that is like basically it's a strong bonding that basically it binds with it. Okay, and at the same time cohesion happens is that when other water molecules binds with the another water molecule, and that is called as cohesion. So cohesion is between water molecule and water molecule together, but unfortunately you won't be able to see one water molecule like this, another water molecule set above like that. Typically it will start to mix together into one that sort of droplet. So that's a cohesion. Adhesion is then on the wall, basically. Okay. Uh, so also water molecules go upside and tight by its walls and close it. Yes, something like that. And it's all by the transpiration pool. The keyword is the transpiration pool. Mm. Keyword is the transpiration pool. Okay. Then translocation, right? All right. Let's it's always from source to sink. So this is a simple word. Source is like where it's being produced. Since we know that amino acids is derived from nitrate ions, right? These nitrate ions come from the soil. So once the is being active transported in, okay, or maybe it's entering the root hair cell, then it has to start move up to the rest of the plant because the rest of the plant also needs the proteins. So this is gonna what happens. It's coming from the source to the sink. The source is the one that makes it. The sink is the one where it reaches to its final destination. So source I always say is from the production center, production or the manufacture center. Manufacturer lah, okay, manufacturer. And the sink is where its final destination is. That is where it's trying to go. Okay, all right. Exactly. Okay, all right. Shall we explain the heart? Okay, but first of all, let me see if there are any questions. Okay. Uh, nearsighted, blood sugar level drop. These are all later. Oh my goodness. Aiya, GG lah. Antibiotic. Oh my god, I still got a lot to go. Can go up one more time. Okay, which one? Which part? This? Yeah. Nothing much, nothing much. Eh, why am I regretting his deal? I'm not regretting anything. Later on, this PDF will be shared. Don't worry. Don't worry, guys. Don't worry. All right. Okay. Let's go to chapter 9, the heart. So, first of all, please remember the labelings. Okay. So, all right. Okay. Something to tell you to ease your mind, right? Do not need to remember tricuspid, and this aortic valve. You can just remember as the atrioventricular valve, and the aortic as the semilunar valve. So this applies for both the left and the right. Okay. By the way, I also want to study medicine. By the way, so but then I don't not interested in study cardiac. By the way, okay, come back. So this applies to both the left and the right. So when we see in this way, right now you are looking at my screen, right? This side is the right hand side. This side is the left hand side. Please remember that. Do not confuse it the other way around. And of course, there's one more thing that you can see is that the left ventricle is very, very thick. When it's very, very thick, you know this is the left hand side already. Okay, so important. All right. Then know all your labelings. Okay, labelings very important. Do not confuse with the pulmonary artery and the pulmonary vein. Pulmonary artery is going to carry that deoxygenated blood. It's the only artery that carries the deoxygenated blood. And the pulmonary vein is the only vein that carries the oxygenated blood. Okay, so the pulmonary circuit is to the lungs, and the systemic circuit is to the body. Okay, just remember. So okay, what are the things that you need to know? Important point is just to remember the labelings. Very important because they definitely can ask you. And then one more thing, the septum. It separates the left hand side of the heart and also the right hand side of the heart. Okay, so this is to prevent the oxygenated blood from mixing with the deoxygenated blood together. Yes, we will go through adaptations later on. Okay, don't worry so much. Okay, and then we have the, you know, uh, oh, this is sinoatrial node. A level stuff. A level stuff. You want me to explain? Do you want me to explain sinoatrial node? No need, right? No need, right? I can explain, but no need. No need. I don't want to fill your brain with some unnecessary stuff. Go and face it in A levels. Okay, let's move on. Now we would they ask what's a septum? No. They will either ask you to label or ask you the function of the septum. What is the function of it? So this one don't really have much, but then just remember that they can ask you what's the function of the valves in the heart, what's the function of the septum. Um, yes, that's pretty much it. And then of course this is the inner surface, inner thing about the heart, right? So there's also the what you call as the coronary artery. So very important. Coronary artery is here. You cannot see the coronary arteries inside because it's on the outside of it. It's like outside of the it's like integrated into the muscle. Is septum like a wall? No. Yes, it is. It's like a barrier, I would say, a barrier that separates the left hand side and the right hand side. You know. Mm. Okay. Okay. Now, functioning of the heart, which is a bit of the how the blood moves from here and there. But I will make it very, very simple. Okay. So first of all, the valves, right? Okay. Not to say the valves first of all, is that the valves is always okay. One opens, another one closes. Very important. There, for in your IGCSE, you just need to know that there's always one side that opens the valve and one side has the valve is closed. And secondly, the muscles in the heart, right? They work the antagonistic. meaning one side will contract, the another side will relax. Okay, so how does this gonna work? All right, okay, I draw this easier diagram. Hmm, okay, something like that. I don't put, I just put like this. This to the lungs. And this one will go in here. Okay, sorry. Okay, very. So this is a heart that I used to first remember the left and the right hand side back when I'm studying this heart. So left hand side is here, right hand side is here, but of course the left hand side is supposed to be thicker a little bit to show that that's the left ventricle. So there's four chambers in the heart. One is the right atrium, this is the uh, right ventricle, this is the left atrium, this is the left ventricle. Okay, so okay, now let's see how the valves open and how the valves closes. So when the blood right flows into the from the body to the atrium, basically the blood is here. The atrium will now contract. So when the atrium contracts, right, the atria contracts. Okay, I put AV valve means atrioventricular valve. Okay, do note. So the AV valve will open. So when the atrium right starts to contract, you just think of that contraction helps to push the blood down. So it pushes the blood down to the um this right ventricle. It starts to fill up. At the same time, the valve is open here. Okay, so the AV valve will open. That is the loop sound. So it's always a loop doop that type. So the loop sound begins with um the AV valve opening. Okay, now I have a lot of blood in the ventricles now, and I want to send it to the rest of the body. So how am I going to do? Is that now the ventricles will contract. So when the ventricle contracts, the now I write SV valve. SV valve opens. AV valve closes. See, one opens, another one closes. It's always like that. One opens, another one closes. So the SV valve open, the semilunar valve will open here to allow blood to eject out to the rest of the body. So it has to go. Difference between bicuspid and tricuspid valve. You do not need to know this, but then if you want to explain, sure. Tricuspid, right? Okay, can you see? Tricuspid is on the right hand side. Bicuspid is on the left hand side. The reason why is tricuspid, it means that there's three cusps, okay, three cusps of it on the atrioventricular valve. The bicuspid valve will have two of the cusp shape. Cusp shape is like that, CUSP. Okay, it has that two cusp shape. But don't worry, do not need to write bicuspid and tricuspid. Just remember atrioventricular valve. That's enough. Okay. All right. So is this understood? Very, very important thing they can ask you in the MCQ or maybe in the paper 4 sometimes. Okay. Where was the semilunar located here? Can you see? Um, this one. All right. Okay, now let's go to arteries, veins, and the capillaries. So, how can I remember this adaptation? I will introduce you my three important structural adaptation of arteries, veins, and capillaries. Just remember these three: wall, valves, and lumen. That's it. You remember these three? So simple. You can definitely score full marks in this kind of question. So the wall of the artery, it's thick. Okay, it has a thicker valve, so the not thicker valve. Yes, the artery has a thick wall, so that it can pump blood in the higher pressure to the rest of the throughout the body. The veins will have a thin wall, I would say. And then the capillaries have a one cell thick wall. Okay, very simple. All right. Then we have the valves. So there is no valve present at the arteries. Oh wait, let me have a look. Sorry. So just need to know semilunar atrioventricular. Oh, okay. Aortic valve is the same as semilunar valve. Take note. Aortic valve is the same as semilunar valve. All right. Okay. And then veins, they have valves. Is to prevent the backflow of blood. Please remember this. Prevents the backflow of blood. Capillaries have no valves. The lumen, the arteries have a narrow, I would say not to say narrow, yeah, narrow lumen. The veins have a large lumen, and the capillaries have an extremely narrow lumen. It's at the smallest lumen. What? Glycolysis, no need to know, by the way. This is in A2 level. A2 level, A2 level. Please don't need to add so much info here. Okay. Used to be an IB. Oh, yeah, really? I see. Arteries, the lumen narrow because of the elastic tissue. Yes, that's right. All right. So memorize this. Very easy. If you cannot remember what to write, wall, valves, lumen. Talk about these three. They are very useful for the structural adaptation of the arteries, veins, and capillaries. Okay? Take note, very important. Okay. Then, as part of the, can you explain why the lumens are the way they are? Okay. The arteries have a narrow lumen because it has already have a thick smooth muscle. The thick smooth muscles helps to pump the blood out in higher pressure. So they do not really need to have a they do not need to care whether they need to have a very large lumen or what, because the blood will still flow at a very very high pressure. For veins, okay, we have a large lumen now because blood has to be sent back into the what do you call as back to the heart, basically. And then there is no pressure in the blood, no pressure in the veins anymore, so they do not have the smooth muscles, so therefore they need a larger space so that the blood can all be sent back to the heart. For the capillaries is joined between the, like, basically got so many of these. This is the capillaries and this is the veins. It's like joining between them. It helps to connect the arteries and veins. So the capillary, right? They just need to have a very very small lumen because they undergo zero pressure, and then they just only need to transport from one side to another only. Because this capillary is important for this respiration of the tissues. So that's pretty much it. Okay. So can you talk about the difference? Okay, it's here, by the way. It's here. It's here. So the structural adaptation, I want you to remember three things. If you think of these three ideas in the exam: walls, valves, and the lumen. Flexibility of the arteries. Flexor, can you give more context? Flexibility of the arteries. Flexibility is not a structural adaptation, it's more of a characteristics already. I'm now talking about structural adaptation, like the way they recoil when this is more of a characteristics. So this will not be accepted as an answer as a structural adaptation. Have to be very careful. Okay? Just don't worry so much. Just remember this three. It's been very, very useful for me. Explain the valves part. Okay, arteries no valves because it's been pumped at higher pressure. Okay, veins, they need to have valves. You know why? Because our leg, example, this is our legs, right? Okay, please don't talk about feet, please. Okay, so this is the veins. So the blood, right, after they have undergo this respiration and everything, so this thing will go up back to the heart. So there is no pressure in the veins. No pressure. And now the veins have to go up against gravity. So we have to go against gravity. The muscles will now help to pump the blood, and the valves needs to prevent the blood from falling down, because we don't want the blood to fall back down. Everything has to go back to the heart. That's the key role of the vein. It's just to send back the blood back um, to the heart. It's measure of smooth muscles and elastic. Yes, it's a structural characteristics. It's just that if you don't want to memorize so much, just memorize wall, the valves, and the lumen. That's it. Okay. So as part of the new syllabus, right, there's an introduction of the hepatic portal vein. Hepatic portal vein is between the GI tract, means whatever the small intestine has already done, the capillaries, right? It like for example, the villi, after it's been absorbed a particular nutrient, it has to go back to the liver. So it has to go by this hepatic portal vein. The portal is linking the GI tract to the liver, so the liver can undergo the whatever the amination, all these. Okay. Mm. All right. Okay. GI tract or gastrointestinal tract. Basically the entire digestive system. You learn the alimentary canal. That's the GI tract. How much you need to know about that? Which one? This one. Just just know what is a hepatic portal vein. Just need to know what's a hepatic portal vein. For the hepatic and artery and the hepatic vein is the same thing as renal vein, renal artery. You see this in the excretion. Carries blood to the liver and carries the blood away from the liver. Yes, that's it. Correct. That's it only. Okay, don't need to. Hepatic portal vein. Yes, correct. And any parts of the body where it needs to go back to needs to go to the liver for the breakdown, then yes, it will undergo this part. Okay, correct. Do I need to explain blood clotting? Do you guys know what's blood clotting? I think it's not very difficult, right? No need. Yes, ah. Okay, never mind. Very simple. Blood clotting is from fibrinogen to fibrin, and they will form a mesh, and then this will stop. And it's all worked by the platelets. All right, very simple. Okay. Okay, now chapter 10. What converts the fibrinogen to fibrin? Uh, there's a there's a very long story behind this, but then do you want to know that? Don't need, right? Hmm. Because the when the platelets are activated, they will release something called as the clotting factor. The clotting factor, yes, is something like a net concept. Clotting factor is that they help to stimulate the prothrombin to thrombin, and then the fibrinogen to the fibrin. Then they form the mesh that which stops the blood from bleeding again. Okay, that's it. All right, let's move on. Okay, now antibody and antigen. For antibody and antigen, antigen can be present on the surface of the pathogen. Very important. And now let's link back to enzymes. So remember, enzymes has to be exactly complementary to the substrate it's trying to bind to. So this is the same exact concept. The antigen will be the substrate, and the antibody will be the enzyme. Again, antibodies is also a protein, so everything needs to be specific. So in order um, for this killing of the pathogens of all these, then they have to bind with it. Specific antigen binding site. A specific antigen binding site to the antigen receptors like that. Okay, so very simple. I don't think I need to go too much on this. Okay, for this I no need to know this. Natural, artificial. Okay, all right. So for active immunity, I think you know one of it is the vaccination, where you inject the dead pathogens. Okay, when you inject the dead pathogen, this is called as the first response of the active immunity. So it helps to create that for you to produce the memory cells. So key thing, memory cells are produced here. Okay, But passive immunity, there'll be no memory cells. And of course, that natural passive immunity is from the mother. When we do the breastfeeding, the breast milk contains that antibodies which are very useful for the fetus. Okay? All right, that's it. Do I need to explain cholera? Cholera? Yes, okay, let's go to cholera. Okay, just make it cholera very simple, by the way. Okay, cholera is consists of is a bacteria. All right. First of all, it's a bacteria. So this bacteria. Oh, can I explain phagocytes? Or do you want me to explain? Okay. So phagocytes, it looks something like this, and this is a lymphocyte. It has a big nucleus. Phagocyte, they will undergo the phagocytosis. Lymphocyte, they will produce the antibody. Is this a good summary? I hope you understand this, by the way. The phagocyte looks better, not gonna lie. uh, it has more nucleus, of course. Okay, it has more nucleus, which are memory cells also lymphocytes. Uh, are memory cells? No, 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 no. It will undergo a specific immune response where they will produce this. They will differentiate first of all. Like any other cell, they will differentiate into some other two cells, and that particular cell will produce the memory cells. So mathematics we have differentiation, and biology we also have differentiation. But don't worry so much. Differentiation, you learn this in A levels. Okay. Okay. All right. Excellent. So phagocytosis is like phagocytes eating up. Yes, absolutely. So I would say phagocyte is almost like a monster eating, okay, like a monster creature. So if they see a foreign material, they just that's it. Lymphocyte, they just act as like the how would you say as the pussy cat that release the antibodies. Doesn't do anything one. Useful, but then it's a pussy. Phagocyte is the real soldiers in our immune system. Without them, we might have got thousands of disease in our body. So let's appreciate the phagocytes, by the way. So lymphocytes, I always call it pussy because they only release the antibodies out only. All right. Okay. If they ask how the body defends against wait. If they ask how the body defends against pathogen, do we talk about lymphocytes scanning the pathogen and creating? It's always the phagocyte that that helps to signal them. The phagocyte will first signal. Let me have a think. Oh, sorry. Yes, the antibodies will first bind with the antigen. Then after it has been infected, then after it has already been almost like um partially killed already, then the phagocytes will come and engulf it. Yes. So the antibody is key. Mm. So do you know why I always love to use so many analogies for my bio? Because I try not to memorize a lot. To me, do not memor I try to limit my memorization. So I use these different kinds of analogies. If I understand what is it, right, I can just think of put this in different situations so that it makes me to understand. Hmm. Okay. All right. Okay, let's move on. So cholera. Uh, so cholera is a bacteria. Right? It's a bacteria. So they will secrete the chloride. They will secrete the chloride ion. So coming back to the osmosis, right? Chloride ions is almost like a salt. So when there's a lot of chloride ions, what happens is that the solute concentration will increase. But before that happens, it has to produce the toxin and secrete them into the small intestine. So let's take it. Yes, this is the small intestine. So they will secrete. They will secrete a lot of these. So what happens that after they secrete, right, then it will cause the decrease in the osmotic movement of water into the gut. So what does this tell you is that they prevent the reabsorption of it already. Wait, if this is 5 marks, what do we need to mention? We 5 marks. 5 marks is a lot for this kind of question though. 3 to 4 marks maybe for that phagocyte and lymphocyte. How do they react to that pathogen? Just mention how the antibodies first binds exactly complementary to the antigen of the this one. Then after they have already partially destroy the cell, then they will ask the phagocytes to to eat up. Okay, undergo the phagocytosis. Okay, there's so many. Okay, so I think you guys understand, right? So what happens is that they secrete the chloride ions into the lumen of the small intestine. Then, of course, due to that so much of that chloride ion in the lumen, it already increases the solute concentration. When you increase the solute concentration, this decrease the water potential. So coming back to osmosis, all of the water will move out. Okay, they will all move out from the body, and it will not be reabsorbed back. So what happens if all of the water is not being reabsorbed back in the body? This will cause the diarrhea. So the diarrhea is the watery feces, where the water is all coming from due to there is no reabsorption of the water back into the body. Very important because water to us, 70% of our body is made out of water. So we cannot have too much of water being released. Okay? Important. How does one kill water potential? Not to say kill, but then it's like the change in the gradient. I would say is that when one side has you feel thirsty when infected. Yes, exactly, correct. Because all of the water has been reabsorbed. Not the reabsorbed. All has been passed out to the feces. So it goes to the large intestine. Um, it affects the concentration gradient. Yes, yes, you are right. So there's a lot of water going to pass through the large intestine. This will decrease the permeability of the walls lah. Okay, so unable to reabsorb back the water, then it just release out as in just makes our pool watery basically. Oh yeah, fun fact. Let me just tell you one small thing that the large intestine, right? There is it's not us doing the digestion anymore, by the way. No more digestion happening in the large intestine. You know what organism that do the things in the large intestine is the bacteria, by the way. So we have many bacteria in our good bacteria, by the way, that happens in the large intestine that helps to do the last breakdown of the nutrients, and then whatever the bacteria release, the vitamins, the water, the salts, they are all reabsorbed back into the body basically. So that is one key thing. So have you ever wondered why there are so many people who got colon cancer? Colon is like the large intestine basically. So it's because of the bad bacteria that lays on some of the parts of our large intestine. So because of that the living bacteria there, right? Of course, when we damage our large intestine, this may cause a lot of infection. So bacteria they can colonize the region one. So like I say, the large intestine looks almost like this, right? Sorry, bad drawing again. So I would say that each bacteria almost like an empire like that. They each part have different types of bacteria one, and they just try to colonize as much. Yes. So bacteria itself has its own So cholera is the disease and bacteria is the vibrio. Correct. Excellent, correct. I saw there's a one question. Where's the one question? Can you explain why antibiotic won't work against viruses? Oh, okay. So, this is in drugs, by the way. Can I come to that later? I come to that later, Ken? Later on, Ayo. All right, let's move on. Okay, so breathing, very important. This process. So inhalation, the external intercostal muscle will contract. This will, the internal coastal muscles will relax. This will contract. Diaphragm will contract. The ribs will move up and out. This will move down and in. This will relax. The volume in the lungs will increase. The pressure decreases. The atmospheric pressure is higher. And this will be decrease, increase. Okay, first of all, let's I want to try to, if you never take physics, right? Okay, pressure is inversely proportional to inversely proportional to volume. Okay, so um, anybody take physics here? Anybody take physics? Okay, I use a I for those who take physics, I will use this concept so that you can understand better here. Boyle's law. Okay, when the pressure increases in one side, the volume will decrease always. So when one side pressure increases, the volume decreases on the another side. Same thing for P2 V2. The pressure increases, the volume decreases. When the pressure drops, the volume increases. All right. So that is the concept. So Boyle's law very good to apply here actually for if you are confused. Okay. So come back. Why does the volume in our lung will increase? Because the atmospheric pressure has a lot of gas particles. So a lot of gas particles, right? It means that it has a higher concentration gradient. When it has a higher concentration gradient, what happens is that the pressure is very high. It will go up. It will basically rush down into our lungs. That's why we must have lower pressure in our lungs so that the outside pressure, which is very, very high, will rush inside to our body. Therefore, inspiration occurs. Okay, inspiration occurs. So the pressure in the lung must be low. Okay, but if it's expiration, the pressure in the lung increases. Why? Because there is so many of the carbon dioxide being in our lungs now. It has to excrete out. So the pressure inside the lungs is very, very high. So the atmospheric pressure must somehow be lower than the pressure in the lungs. Then only the diffusion can occur, the exhalation can occur, going from higher pressure to lower pressure. Okay, uh, okay. How do I explain the intercostal muscle? All right. In our ribs, I need to find a diagram with this, right? Okay. Basically, it has something like that. Can you imagine this? Okay. This is the external. Okay, wait. Let me. This is the external. This is the internal. External is thicker. Hmm. Typically, external means it's on the outside basically, and internal is on the inside. Hmm. So external intercostal muscle will definitely contract during inhalation, so that the muscle right contracts, right. It allows the diaphragm to move up. Okay. To move up, the ribs are also up and then out, something like that. Okay. Okay. So I just think that the atmospheric pressure is at the speed of air. Yeah, something like that because it's always from higher pressure to lower pressure. Okay. Remember, higher pressure to lower pressure. This is something like diffusion, by the way. Always from the higher concentration gradient to the lower concentration gradient. All right. Okay, and one more thing. Please memorize this. Memorize this table, by the way. Oxygen in inspired air has 21%. expired air have 16%. If you learn in chemistry, right, the environmental chemistry, you know that 76% of the air is nitrogen, so 21% is from oxygen. Okay, the carbon dioxide is at 0.04%, and 4% is expired air because we are exhaling more of the carbon dioxide out. Nitrogen always 76, 78%. Water vapor is lower, and water vapor is higher in expiration because more air is being used. More of the water vapor, the gaseous state, is being released out. Okay, what about the thorax volume? Our thorax volume is same as the lung, by the way. Thorax is part of our respiratory system. So, um, same thing. Thorax volume, it has to be higher in the inhalation. Okay, so that the air can come in. That's why we have the cartilage, by the way. Remember cartilage? Cartilage is a C-shaped ring. That C-shaped ring prevents the collapse of the entire trachea and allows for expansion. Because when there's more air coming in, right, the trachea has to, it can be stretched almost like a coil like that. So it can expand basically. All right. Okay, can we go to deamine? And why doesn't nitrogen increase? Oh, because in the atmosphere it's always the same. We don't it's always the same. But of course this thing, right, is when the air condition, right, is at the very very best condition. Of course, we of course do not breathe 21% of the air anymore. If you're in the capital city of a particular location, you won't be breathing 21% of oxygen in. You get what I mean? So this kind of results, right, is all from the when the condition that is the best highest one. Okay. Okay. Uh, any questions, by the way, before I go to deamination? Any questions? Any questions? I hope you learned a lot today, by the way. Oh, I'm so about to die soon, maybe. Still not even halfway yet. Brain fried, but I learned a lot. Awesome, at least. I know I'm supposed to do this like much earlier on, but then because I also got a lot of stuff to do, so that's the reason why. Um, hmm, okay, shall we proceed? Shall we proceed? Okay. So, what time you should sleep in during exam? I'm a morning person, so I sleep very early, by the way. So, in my local time, I sleep at when I'm doing my IGCSE exams, I slept at like 10 or 10:30. 10 to 10:30. I can go and sleep already. I don't sleep past 12 o'clock. Although a lot of my friends sleep at 2 a.m., 4 a.m., I say that I don't do that. So sorry, by the way, because no, now it's not. Now I just finished my exams, by the way, so I can stay up just to support you guys. Okay, that's of course giving back. Okay, so all right, let's move on to deamination, guys. Okay, so excretion. Am I in college? Yes, I am right now. So deamination is the process of removing the nitrogen containing amino acid. So you know that a protein contains carbon, hydrogen, oxygen, and the nitrogen, right? So the key point of deamination is to remove that nitrogen containing part. All right, is to remove that thing out and convert it into urea. So this urea, right, is going to be sent to the uh, kidney. Ah, I, okay. You know why? Because when I'm in international school, right, I technically skip 1.5 of my original age. If you're curious, I skip half one, not because I'm intelligent. It's by my birth date. Okay. Let me do this. Okay. Let's continue. You're welcome. You're welcome. Okay. Okay, now let's go to the kidney, nephron. So please remember that you need to know the labelings of the kidney, like the cortex, the medulla, the ureter, all right, and the renal pelvis. Please don't confuse the ureter with the urethra. The ureter is the kidney tube that is going to the bladder. Urethra is in the penis that carries the urine and the semen. Okay? So do not confuse with these two. Okay? Alright. When it comes to this nephron, right? So it starts off, okay, whatever comes into the glomerulus is at very high pressure. So these glomerulus, right, they will accept all of the nutrients in a very high pressure. Because right in the how many Malaysians in the chat? Yeah, I'm in Malaysia by the way. Okay, so I think you guys know I'm from Malaysia. All right, so glomerulus, they will undergo this ultrafiltration. So what does ultrafiltration means? It means that whatever we have right in the um basically in the nutrients, everything is gonna be filtered, and what they will filter out is all the larger molecules. So like proteins, okay, uh, ultrafiltration, it was in the glomerulus first. Glomerulus first. Okay, so because the glomerulus has a lot of pores, a lot of pores. So in order for that to penetrate into the Bowman's capsule, right, it has to undergo the filtration first. So this filtration, right, they will basically filter everything out. They will filter all the protein, the red blood cell, the white blood cell, only to allow those more soluble things like the urea, the salts, the water, everything can only, yes, correct. Ultra means big. So it's called big filtration. So you can remember this as the filtration of large molecules. Yes, that's correct. Ultra is the word of big. So that explains why. So that's the ultra filtration. Now we are going to undergo the renal tubule, or this is the proximal convoluted tubule. You just need to know what is this tubule. That's enough. So I still don't get what the Bowman's capsule. The Bowman's capsule is just basically like a capsule. It's like a storage thing that it receives from the glomerulus after it's being filtered. Right. That in chemistry you have the filtration. This is the filter funnel. This is the residue. This is the filtrate. So what the Bowman's capsule does is that it receives the filtrate. The residue means it's all the large big molecules that cannot pass through will all be sent back to the blood vessel. So that is the key thing. The Bowman capsule will help to capture the filtrate. So proteins, red blood cell, white blood cell, all those things that these inhibited molecules cannot come in. But glucose can enter the glomerulus. Please remember, the glucose can enter the glomerulus. Okay, sure. So, what cannot go through is the protein, red blood cell, white blood cell. Sometimes can be hormones because hormones needs to be broken down in the what's that called in the liver. Okay, if they need to be broken down. For what is being accepted in is the salts, the glucose, the amino acids, and the urea. Okay. All right. Now we enter the proximal tubule. Proximal tubule is where the selective reabsorption happens. Okay, selective reabsorption is where the tubule will be basically a bit picky, I would say. They will be picky on choosing which materials to be absorbed, and that being really really like what do you call that, as being very selective is what we always absorb the valuable substances. So For example, like glucose, they are all very valuable substance. The body needs it because glucose is an important reactant for cellular respiration. So that's very important. Okay, remember guys, I will send the PDF later on until where everybody brain got fried already, and then we probably, let's see, if I have another time, I will do another continuous session for you guys again. Okay, so the proximal tubule will reabsorb the glucose. That is the key point. Okay, they will reabsorb the glucose. Okay, so why, for a normal person, glucose is not supposed to be in the urine, because that person means it has diabetes already. So when the glucose is being reabsorbed, it's going to go back to the blood vessel and then goes back to the rest of the body. So that is the key thing for selective reabsorption. Okay, then you go to the loop of Henle, then you go to the distal tube, then the distal convoluted tubule, then to the collecting duct. Collecting duct is where all the urine is formed already. And then the urine will go to the ureter. So ureter is the thing, the tube that links from the kidney to the bladder. Please, it's not urethra, not urethra. I know I keep on saying repeat, but I hopefully nobody uh does this by writing urethra. Okay, so uh, so how do questions asked in excretion is, they probably might ask you to explain a 6 marks question, maybe a 4 to 5 mark questions on explaining how this nephron works like that. Does glucose get reabsorbed through the villi? Yes. The loop of Henle will basically make the urine more concentrated, I would say. Urine a bit more concentrated. Okay, because what happens in the loop of Henle, right, is that some parts of the wall, it is permeable to a particular substances. You do not need to know much for this IGCSE. If you ever interested with studying further with bio, you should learn this in the future. So just to clarify, the kidney consists of medulla, cortex, the renal pelvis, the renal vein, the renal arteries, and also the ureter. Okay, that's the key labelings you should know. So the cortex, I always remember this way. You know, the cortex, right, is the C-shaped one. So cortex has a C. Cortex is the, yeah, this part. Okay, so let me just do the labeling. Okay, so the cortex is here. Okay, cortex. This is the medulla. This is the pelvis. And this is the ureter. Okay, all right. So do you understand? Wait, does the small soluble? Yes, some salts, water, they are all pass. So medulla is where the nephron is located. Yes. Partially the nephron. The nephron is partially in the medulla and also in the cortex also. It's in both. It's the loop of Henle, right? Okay. Basically, what I would do is that I will put in this. I put this as called as the intersect. Put this as a separation. The top part is the cortex. The bottom part is the medulla. So this loop of Henle will stretch down to the um to the medulla basically. Okay. So do you understand this now? Proximal convoluted tube reabsorb the glucose. Okay, by the selective reabsorption. Glucose, amino acids, waters are all also being reabsorbed. So, all right. So, any questions? Any more questions before we move on? Next part is coordination and response. All right. Awesome. What are the key points of answering the question? Depends on the question. So, like what I've mentioned. All right. Thank you so much for joining. Okay. The key points of answering this kind of question is just to mention the high pressure first of all, the ultrafiltration, the selective reabsorption, and also the final one. Okay. When will the lecture? Not a lecture. Please, not a lecture. This is a revision session. Okay. Um. Okay. I want the adaptation for tubules for absorption. Adaptation you don't need to know much. I meant like no. Hopefully, I try to. Should I stop where? Well, I know I still got a lot though. Okay, can I do one more chapter to chapter 40? Then I will stop here. I will stop there. Sure. I've been hold on, one more chapter to go. No problem. You're welcome. Oh, you want me to stop here? I will try to do one more session tomorrow. Can you can you guys please make sure you are ready with your notifications at 8 p.m. Same thing. Same timing as today. Okay. And yeah, we will continue the rest of it. We will answer the questions and answers. We do tomorrow. 8 tomorrow. Yeah, I will try to do tomorrow. Same time? Yes, same time. Time zone GMT plus 8 for my time. GMT plus 8, 8 p.m. Okay. I will send the PDF document into the where do you want me to send this? Should I send this in the you know when you go to my IG bio IG biology or in the in what's that called the thread in the study session? I think I will send both. IG biology. Okay, sure. I will send both lah. Okay. So all right. Thank you so much for joining. I really appreciate you spending your time here because I know biology isn't for everybody, and sometimes I just want you to understand more about yourself. Most importantly, don't just do bio just for the exams only. Sometimes it's very useful to know more about yourself also. So we will continue everything tomorrow. So thank you so much for joining. Arigato gozaimashita. Terima kasih. 谢谢你. Okay.